So I'm Bob Murphy. I'm going to tell you about our work with Cell Organizer. And to get started, I'm going to try to set the stage for why we are building Cell Organizer and have been for many years. One of the reasons relates to this. If we want to do cell simulations, then we clearly need to have spatially and spatiotemporally realistic geometries of cells that reflect where all the components of the cells are, organelles, proteins, other macromolecules, in order to do those simulations. And that means that we naturally think of using some kind of microscopy, especially fluorescence microscopy, to sort of try to collect information about where things are within cells in order to try to put that together such that we could eventually be able to try to simulate in a spatially realistic way, maybe a whole cell process. But the question is, how do we get geometries that are accurate for many, many different parts of a cell? And this is especially a limitation now because there are lots of good systems now for building cell simulations that can incorporate spatially realistic geometries and so on. But the vast majority of the projects that are done using tools like that, which you've been hearing about for the last 2 days, use geometries that are either hand-segmented from electron microscope images, which is an incredibly painful process, or are drawn by hand, made up by drawing a bunch of things with a drawing tool. And those things dramatically limit the number of geometries that we have available to do spatial cell simulations. All right? And for example, when we do something like this, where we're doing hand-segmented images, we get no idea of the effect of variation from cell to cell because you're doing one or a handful of cells by hand, and therefore you're really not getting any sense of that variation. And that's where cell organizer models can come in. Now, for those of you who are not that familiar with fluorescence microscopy, part of the point here is that when we think of doing cell simulations, we typically think like doing a simulation in a cartoon like this, okay? And we might even think of taking such a cartoon and drawing one ourselves. And the problem is this is not what a cell actually looks like, not even close. All right. Here is an example of an actual image of the endoplasmic reticulum, which isn't too far off from that, but significantly. Here's a distribution of lysosomes within the cell. Notice they are much smaller than they are drawn in this geometry. Right? The ER was about to scale, but the lysosomes are way off from scale. Here's the distribution of microtubules. Here is the distribution of mitochondria. Notice also these things are huge compared to what they actually are in the cell.
What kind of cell?
These are 3T3 cells. And so when we take images of cells like that, traditionally what we have done is to take them and convert those images into some sort of conceptual understanding of what is being depicted in the image. And that's usually been done visually. And for example, to take, this is an image of a lysosomal protein, to look at that and say, oh, well, lysosomes seem to be sort of medium, round, small things, and they're kind of distributed maybe a little more towards the nucleus. That's the kind of thing that people have been doing for decades of cell biology. And the problem is, of course, that is very, very incomplete and very painstaking to do. It's all done visually. Now, beginning about 20 years ago, we started to apply what's called supervised machine learning designing features to capture these patterns and using those features to classify different organelles and so on and so forth. Okay? So there's a huge amount of work now on using numerical features to describe images and then classifying them to say, for example, this is healthy, this is cancerous, this is lysosomes, this is endosomes, whatever it might be. And that has also been incorporated into so-called high-content screening or high-content analysis, which is systems that often involve automated microscopes that can take millions of pictures and then automatically analyze them. And the way in which that is done is largely to take images of two different conditions, say normal and cancerous, or unperturbed and the perturbed thing that you're looking for, or anything. It doesn't have to be two, but very often it is. And you take example images of those two things, calculate features from them, and then try to figure out how to use those features to tell the difference between those two things. So that's a very standard thing to do nowadays. The problem with that is that these features are descriptors of the cell that very often are specific to the type of microscope, the type of conditions that are used in order to collect the image. Things like texture features are very widely used, and texture features are exquisitely sensitive to the pixel size and the bit depth and so on and so forth, right? So the problem is, if here in Pittsburgh we do an experiment and we learn a system that can tell the difference between A and B, and then somebody in California wants to use it, unless they have exactly the same microscope and exactly everything, they can't. All right? Because those features are not comparable, and obviously anything based on those features are not comparable. That means they would have to do their own experiment and then train their own thing, and then we can't exchange between us. So if we say these two things are different and they try to do their experiment themselves, they might say, no, they're not different. Right? Because under our conditions, they don't seem to be different, or they're different in a different way. So what we're trying to get to for all of this is that approaches that are looking at images and then trying to either extract words or extract features, that extraction process is focused on making decisions, discriminating between different possibilities, and it is inherently losing information as we do that. Okay? Because it's extraction. That means take some part of it. Now, in parallel, a lot of cell simulations, which we've been talking about, are based on the idea of a compartmental model, that you define compartments within a cell and you say, okay, there's so much volume of the cell that is this compartment, lysosomes, nucleus, whatever. And again, we can do that with a cartoon. We can do a real image and use it to just calculate areas. Or we can actually take a real image and use partial differential equations to do simulations on it. These are some of the ways in which we can take information from images and try to do compartmental simulations, where each individual part of the cell is considered separately. But many of those are not realistic. So part of what we're trying to do here is to make the case that if what you want to do is tell the difference between normal and cancer, or between endosomes and lysosomes, that's a discriminative task, and feature-based systems are okay as long as you don't want to compare between different places. But if what you're trying to do is to build a model of a cell that you do simulations of, that's not anywhere near enough. Okay? So if I were trying to model the growth of an apple, it wouldn't be helpful to know that it's red. That's not enough, right? If I want to compare the organization of an apple and an orange, I can tell the difference between an apple and orange by their color, but that doesn't help me put together an apple or an orange. Okay? So that emphasizes that what we need is to be able to take images and build generative models, not discriminative models that just tell things apart, but generative models that can capture essentially all the information in an image that or images that we're working with. Okay? Now, exactly what do I mean by this idea of generative models? So you are all familiar with this concept. When you were kids, you were shown numbers and letters, and you were tasked with learning two things. One, to be able to recognize numbers and letters, and two, to be able to write them. And you were presented with examples, and you sort of had a couple of choices. You could have taken each of those and memorized every 5 you'd ever seen. And if you saw a new 5, you would look and see which, or a new number, you would look and see which of the memorized examples that you had was it closest to, and you'd say, okay, that's a 5. All right? But that's not what you did. Oh, by the way, if you were asked to write a 5, you could just pick one of the ones you memorized and copy it. Okay? But that's not what you did. What you learned is a mental model of what it means to be a 5 and a 3 and a 7. Okay? That model is inherently a statistical generative model because you can generate a new 5 that is not the same as any 5 you've ever seen, but is within the family of all the 5s that you've seen. All right? That's what makes it generative, right? So we generate new instances that we would say are drawn from the underlying model of what it means to be a 5. Now that's a really important concept. Everybody got that? Yeah? Okay. So that's what we're trying to do, is to do that for cells. Okay? We want to take a bunch of images of cells, and we want to build a model, a statistical generative model, of the spatial organization of those cells. Okay? And just to drive that home, we can think of the model that we're trying to build as that statistical model of all of a particular cell type. So let's imagine that we have a plate of HeLa cells, and we trypsinize them into a test tube. And they are all like potential HeLa geometries that could be expressed when we put them on a plate. But we have one in a test tube, and we shake them up, and we pour them out on the test tube. And each one of those cells, when it hits the plate, is going to adopt a geometry in terms of its cell shape, in terms of where all the organelles are relative to each other. Right? And you can think of each one of those cells as making a choice, drawing a number from the statistical model of HeLa cells. Does that concept make sense? So that is the underlying reality. Underlying reality is that there is a model constrained by the biochemistry of the cell from which each of those cells is choosing, okay, when it hits the plate. That's what we want to learn, is that model. Okay? Now, any individual cell draws from that model, and then we can take images of that cell. We can take multiple images of that cell, and we can try to build a model of that particular cell. But we can also do that for many cells and build a model, learn that overall model of a particular cell type. So that's what we're going to talk about. All right, we're going to skip over that. So the work I'm telling you about is all part of this cell organizer project. And the basic idea, as we've talked about, is that we take bunches of images of cells, and we're going to go through a training process where we build models of different components of the cell. And I'll talk more about this. And then from those models, we can synthesize new cells that can, for example, be used as input geometries to systems like M-cell. And our goals are to be able to have that process be automated, that it goes directly from the images. As we said, it has to be generative. It has to be statistically accurate, reflect as much of the variation from cell to cell as we can. And here's an important concept. We want it to be compact. All right? We want it to not just be taking all of the bits of all of the images and putting them into some kind of model. Right? That's not a model. That's just the data set. Right? So it needs to be smaller than the sum of all the pixels that it was built from. Okay? Does that last aspect make sense? Right? Okay. Okay, so how do we try to do that? What we're going to do is we're going to take our images, and for each image, we're going to extract parameters, or we're going to parameterize that image. And each of those parameters are going to capture something about that cell or about the distribution of an organelle in that cell, a structure, whatever it might be. So this part is going to be very generic. So each parameter gets extracted from each cell. That's what all these arrows mean. So from cell number 1, we have a list of parameters that describe it. Now, here's the key, because this sounds like I'm calculating features. I call them parameters rather than features because they are supposed to be, they are designed to be such that I can reconstruct the image from the parameters. I can't do that with features. So if I tell you, that I have this texture feature, I won't get back. If you give me the 10 texture features for a cell, I won't be able to generate that cell back again. I'll be able to generate other cells, but I can't generate that cell again. Okay? So these parameters, so there's a very, very important concept right here. These parameters are such that they should be able to allow reconstruction of the image to some level of accuracy. They may not be able to reconstruct everything, but to some level of accuracy, they have to be able to reconstruct.
So if I just had one exact image, that was a particular image, one way to parameterize it would be just to do the wavelet transform of it and make a JPEG. Right? That's what a JPEG image is. From all the JPEG parameters, I can reconstruct that exact image.
Yep. So that is a parameterization of an image. Now the question is, and this is what the whole thing is that a useful parameterization of the image? Okay, because when I take another image and do the same process, I don't have any way to correspond that representation, right? Such that I can try to build a model of more than one thing. But there are examples, so that is a very good example of what we're talking about, okay? Does that make sense? So we're taking the image, converting or calculating things from it such that those things should enable us to try to reconstruct the original image. So we do that, however many parameters there are. We do that for lots of cells. So what we end up with is a distribution of each of the parameters across all of the cells. All right, so we have a two-dimensional matrix, you can think of it as, cells, parameters. Okay? And then we build some statistical model from that matrix. Okay? Some model that, given the parameters, will tell us the probability of observing a particular parameter value for one of these parameters, right?
So what does the pipe mean?
That means given, right? So this is the probability of observing any of these parameters given some parameters of the model. Okay, that's what theta is. Okay? Now, this is a generative model because if I learn, so what I'm learning is this theta. That's telling me the distributions of these p's. And if I know that, I can sample statistically from it, just like I can sample from, if I have a model of the distribution of faces of a die, right? If I roll the die and I can generate, I can say, okay, this is a fair die, all of them are going to come up 1/6, or 6s come up twice as often as everything else. Okay?
So P is also a 2D matrix.
So P is,
Oh, big P.
that's the probability. That's the probability of. P is the probability of observing a set of little p values given the parameters of the model. Does that make sense?
And isn't it different for each?
So theta is the thing we learn that enables us to predict the distributions of the parameters that we have just learned from the images. Okay? Images converted to parameters. We have a matrix of many cells, many parameters. Then we learn theta, which is supposed to summarize that distribution. So in a very simple model, for example, we could say every parameter is just represented by a Gaussian. Right? So all the model is, all the theta is the mean and standard deviation of each of the parameters. Right? It's a very, very simple model. Okay? Or,
But then in the middle, I mean, that I get, but I don't get the single little p and the single big P.
so this is P sub I. This is the P means probability here. Probability of
Any p sub I. Any
observing any of the,
got it.
okay?
Now I get it.
And a more complicated model would be a multivariate Gaussian, right? Where I would say it's a mean plus a covariance matrix, okay? Or I can get as complicated as I want with that model. Now, once I have the model, I can sample from the model, get new vectors of the p's, and synthesize images from them, okay? That's the fundamental idea. All right, now, the point of this is that those parameterizations, those parameters, and the models that are built from them are representing the underlying reality of those cells. And that's the same in California and in Pittsburgh. Okay? Does everybody follow that? So that what we're describing is, you know, shape and size and distributions of organelles and so on, not the images that we have of those things, but the parameters that actually generate those distributions. You know, yes?
So, I mean, I guess to be clear, depending on how you do it, you might still be learning features or parameters that are specific to your image type unless you have a diversity of especially if you're going straight from pixel values or something like that.
So there is, yes. So I overstated it in that, for example, if I have a 5 by 5 pixel image, the model I learned won't actually be very good of the cell, okay? So
But another question.
modulus, you know, the resolution issue.
Oh, sorry, I meant even in a full resolution image, depending on what your parameterization is, it still might not be able to distinguish, might still not be able to generate images from different, for example, sources unless those are included in your training.
Well, clearly that's another issue, yes. So for all statistical models, you always assume that you have a sampling of whatever it is that is the reality, right? So again, if I only had 3 images and there are 5 different populations, I'm not going to, you know, have a good model. But so that's absolutely correct.
How do you get
But
the
I-
parameters
but
that'll be general enough?
so there are really 3 questions there. One, resolution. Second, enough cells that you get all populations or whatever variation you want. And the third is the choice of the parameters. And we're going to
How do
talk
you
about
measure? Yeah.
we're going to talk about that extensively, okay? And that is actually the hard part,
Okay.
all right? The rest of this is pretty straightforward. So here's an example already, okay? So there is an image of a 3D, or of a nucleus from a 3D image. And the question is, let's try to make a model of the shape of that thing. So now there's lots of ways that we can do that. But one of those ways is to take this, convert it into what are called cylindrical coordinates, right? So it's just a coordinate transform. And then fit that with a spline surface. And a spline surface has only 32 coefficients, okay? So, and if we add in the height of this thing, then it gives us 33 parameters that we're saying do a reasonable job of estimating the shape of this thing, okay? And that would be an example. Now, you could argue about the choice of whether that's the right model or not, and the issue becomes how well does that model reconstruct or allow the reconstruction of the original image. So here, for example, if we regenerate this nucleus, notice this is smooth, all the little bumps are gone if we regenerate that image. So if we care about those bumps, and the question is, how do I know if I care about them? Well, we'll get to that in a minute. But if I care about them, then I say, oh, that's not a good model. But if I don't care about them, it's a good model, okay? Or it could be. So reconstruction error and whatever the biological thing it is that we're trying to model. So if I can build a model like this and it predicts the properties of the cell reasonably, then it was good. If it doesn't, it wasn't. Yes?
Yeah, I guess just in face value, it seems a little arbitrary, I guess, to choose the most spline surface. So is there any reason why we should have some prior belief that nuclear shapes should be represented as such? And I guess just in general, going forward for the models that you pick, are there reasons prior, or is it sort of try different models and see what works the best?
I think it's largely the latter. Yeah. But we're going to talk about in a second now, and Chantal is going to talk about it later today, is that these versions of models are parametric models where we're making a choice of what the model looks like. There are also non-parametric models that try to avoid that. Okay? So that is closer maybe to what you're looking for. But I think the best way to look at this is that you want to be trading off reconstruction error versus compactness of the statistical distributions that you get versus being able to capture the properties in a simulation that you want, right? So I think those are and that's a kind of a standard thing, all right? Now, another concept that comes up here is that the things as I presented them, I was largely presenting them as independent of each other, and they may not be. So for example, the places where lysosomes might depend on where the microtubules are, or where the nucleus is, or where the ER is. So models of different components may depend upon each other. We're going to talk about that a lot. And we would say that those models are conditional upon each other, and that we're learning the conditional dependencies. Now here's a quick example, which is that a model for cell shape may depend upon a model of nuclear shape or vice versa, right? So like a small nucleus, a big nucleus can't be inside a small cell, so they depend upon each other. All right, so we're going to talk about that. Models of cell shape. Here's an example. What we're going to say is that that cell shape is dependent upon nuclear shape. Now what do we mean by that? What we're going to do is learn a model of how the cell and the nuclear shape relates. And one of the things we could do very simply is to think of, all right, I have a model of the nucleus. Let's say that spline surface model or anything else. What I want to do is capture how the cell shape depends upon that. Well, I could just build a ratio model of the distance from here to here versus the distance from the nucleus to here. Do that over 360 degrees. That would be a ratio model that is capturing that dependency. And that would prevent me getting a nucleus too big for the cell, because I would always be constructing the cell around the nucleus. Okay? So there's an example of generating that way. Now an alternative I just mentioned is so-called diffeomorphic methods, and I won't go into that because Chantal is going to talk a lot about that later. Now, I mentioned vesicular organelles being dependent on each other, being dependent on other things. One of the things they're clearly dependent upon is upon the cell and nuclear shape. So if I start out with a model of cell and nuclear shape, then basically everything else within the cell is going to be dependent upon that, including the positions of organelles, for example. Okay? And how do we try to model that? So here's an example of an image of a lysosomal stain, the lysosomal protein. It's a very noisy image. Here's where I start making my generative model assumptions. I could model this as texture. I could just say lysosomes are just little blobs of fluorescence, and I'll make a texture model of this. But I add in my parametric assumption, which is lysosomes are actually membrane-bound organelles that are sort of roundish. And therefore I ask, what distribution of small roundish things was the most likely to have generated this image? That's the Gaussian mixture model, for example. Okay? So I then just find a set of Gaussians that will generate this with the maximum likelihood. Okay? Does that make sense? Now I have the number of lysosomes, the sizes, and the ratio, the axial ratios, and so on. And I can learn a parametric model from those. All right? Size, shape, intensity, and so on. And that gave me the objects themselves, the lysosomes themselves, their sizes and shapes. I need to now figure out where to put them in the cell. I said it was going to be conditional. So I can do a similar kind of thing to what we did before with the cell surface depending on the nucleus. In this case, I have the position of an object depend on its distance from the nucleus and the distance to the cell, so that I learn the probability that I will get an object at every place relative to the cell in the nucleus. And there's the probability density function that I learn. Okay? So that says this organelle is more likely to be on the right side near the nucleus. Okay? And you notice I oriented this by finding the major axis of the cell. Okay? All right, and so if I put it all together, I can simulate example images of cells and organelles. Now, that emphasized two things: the objects, the size and shapes of the objects, and their spatial distribution. For many objects, they are what we would call puncta in these images, that they don't really have any size or shape. They're just points. In which case, I can skip the first part. And all I do is find where they are, and then just build a model of, find them, and build a model of where they are. Okay? And one of the ways that we can do that is to use what are called point process models, and what we're going to talk about next. So here are example images from the Human Protein Atlas. If you guys don't know about this, it's a tremendous resource. Yes, you had a question.
Something like it has the shape defined, we just don't have the resolution to tell one of them.
Yes, absolutely. And what we're going to emphasize here is differences in the spatial distribution and ignore differences in size and shape. But we can certainly add those in. Right? In other words, the model is going to tell us what's the probability something appears here. We can have a separate model that tells us what it looks like. Okay? And that model could even be learned from a totally different type of image. Yes?
How often does this produce overlap in the output of?
In the probability density functions? Or?
Yeah, or like maybe an instance of the generated.
Like one being the same, yeah.
Like how often does it happen where the nucleus is overlapping with the lysosome?
Oh, I see what you're saying. Okay. So in 3D images, that shouldn't happen, right? Assuming you have reasonable resolution. And you can include an assumption that you're not allowed to be inside if you wish to. It really is a problem when you're using 2D images, because you can't tell what the heck anything is, right? So don't use 2D images, because it's a projection, right? Or, in a confocal, it's still a slab, right? So these are images. You can't really see them too well. See these little green dots? Those are different proteins that have been stained by the project, right? So these are 11 different, yeah. Okay, thank you. Right? 11 different structures stained by various antibodies. Okay? And the question is, can we build models of those things? What we're going to do is we're going to take the original image, and this is one with a lot of dots, and then we're going to segment out all the individual puncta by basically just using high-pass filters, and then take all of the remaining fluorescence, so we have two halves now, the puncta and everything else. And you really can't see them, but there are red microtubules running through this. Okay?
You said 11, but there's only 3 colors.
There's 11 different proteins in different cells. So each one of these were imaged separately.
All right.
Okay? But there are 11 different proteins. That's part of what's going to become the point. Okay? Okay, so we segment out each individual one, and then what we're going to learn is for each puncta, we're going to measure its position with some coordinates, we'll call them relative to some factors. And what we're going to do is we're going to say the frequency with which I observe n of those objects at positions in the image given by x, assuming I know how many I'm supposed to generate. So this overall probability of n objects is given by the product of the probability of each of them occurring at that particular position. Okay? So that's the fundamental structure that we're going to do. And then there's some normalization constant here. Okay? So this is a sort of a basic idea where we're going to now capture everything about the probability of something occurring into this B. Okay? Now, what we're going to do is we're going to construct factors where, and this, I just realized, I've been using this slide for years. That shouldn't be x, that should be f. It's the factor, right? That's the
related to the x in the original slide.
x is the coordinates.
Right? The x and the next x is not that.
This x is the coordinates. This should say f. Okay. The factors are the things that the coordinates are going to depend on. Okay? And so what we're going to do, those factors are going to be things like the distance from the cell boundary, the distance from the nucleus boundary, the distance from the nearest microtubule, and the average microtubule intensity or something like that. And so this B, we're going to build up from these various factors and various parameters. Okay? So what this is going to say is, for example, if F1 is the distance to the cell membrane, and F2 is the distance to the nuclear membrane, then we have some weighting of the distance of one and some weighting of the other to the other, and you put them together, and that gives you the probability that there is something there. Okay? So if it's something that's near the cell membrane, this weight's going to be big, this one's going to be small, and the other way around. Okay? Does that make sense? So that's a basic point process model. And so here are those factors, right? So here's what it means is that each point in the cell, I measure that factor. I have a value of that factor, which is distance. And when I want to calculate the probability of an organelle at that specific spot, I take the value of that factor times its weight, and I add the value of this factor times its weight, and I keep doing that until I get an overall probability. All right, is that clear?
What were these factors chosen?
Another great question, right? So the idea is to try to have something that represents whatever possible input there can be. So the images that we started with were labeled for the nuclear, with a nuclear stain, mitochondria, endoplasmic reticulum, and the unknown protein, or the variable protein. What's that? And cell membrane we can learn basically from the others. We can find the cell membrane from the other stuff, including the autofluorescence. Okay? So the point is, we want to have, in the initial model, we want to have inputs that use as much of that as we can. But also, we want something that is roughly invariant to shape. Right? So that's where distance comes in. But again, parametric choice. Right?
How did you decide a weight? Like
You're going to do fitting. Yes, absolutely. We don't just, we do, right? Well, we choose the form. We choose the factors and the form of the equation. Then we fit. Okay? All right? So what we can then do is take these factors, and you'll notice, for example, that we have distance to microtubules, but we also have density of microtubules. All right? Because distance is a little, requires thresholding to say whether there is a microtubule there or not, whereas density is much more straightforward, right? Just integrating. So the idea was to have a little bit more robust way of putting that in. All right, so what we do then, we take these take a bunch of images, learn the weights, and then evaluate for a test image how well the model predicts the positions in the test image. Now, this was actually the world's smallest data machine learning example, because we had Human Protein Atlas has one image, 2 images of each protein. So one image was the training data, and one image was the test image. Now, each image has hundreds of puncta in it. So it's not like we only had one data, but we had 100 data points, let's say, from the training image. So we could do a reasonable job of learning a probability density. And then we had 100 points that we could compare it against in the image. But it's actually 2 individual images we're using. Okay? So we're going to build those models. Everybody gets that part, right?
So you just hope you didn't pick a weird cell?
Hope they didn't pick a weird cell. Yeah. Yeah. So absolutely. So what we're going to do then is we're going to construct models with different numbers of these factors, different combinations of the factors. So if we build a model just from distance to the nuclear model, the nuclear boundary, we're going to compare it to a model where we include the distance to the cell boundary as well. If we do that, we get an increase in the overall likelihood of the model. Okay? If we take that model and add in microtubules, we get an increase in the likelihood of the model. But if we take that model and add in ER, we actually lose information. That's overfitting, right? It's trying to learn a relationship to ER, but there isn't one. So it puts in weight on that, but that doesn't generalize. So we actually lose. Does everybody get that idea?
Exactly what overfitting is.
Yeah, so okay. So the idea is that from the one instance we had, it seems like there might be a relationship to the ER, but there isn't. So it learns one.
So you calculated something that's useless.
And it wasn't useless for the prediction. Exactly. That's why we have to do this. Yes?
The fitting criteria capture that? Because if you add noise to your system, that shouldn't always ruin your data. A fitting criteria.
But we're not adding noise. We're adding spurious or adventitious information. Right? In that particular cell, it looked like there was a relationship to the ER. But the relationship that it learned didn't turn out to be true for the next cell. Okay, so it's essentially it was noise, right? But it was fitting that. Now remember, we're learning a model on one cell, we're testing it on another. So there was, a coincidental relationship, shall we say, that didn't show up in the test. Sorry, go ahead. You.
So, like generally for non-parametric or even parametric models, if you have a training set on which you train, and then you have a test set on which you test, you cannot go back to the training set and retrain because that would be unfaithful.
But we're not doing that. We're training and then testing on a new image.
So then the only thing different too was that the endoblock, the reticulum was not represented, right? We cannot go back and modify the model.
No.
You say that representation. You say this representation is wrong because it needs the information. I'm not going to keep it in the model.
How do we know which one's right? They didn't train on enough data to get a proper representation of the ER since it's just one image, right?
So then you just stop there because if you do overfitting, then you can basically reproduce whatever you want.
Yeah, my sense is that they can't.
No, so the idea is we're training separate models. Well, a model just of this, and then a model of these two together, and a model of these two plus these two. And we compare the likelihoods on the held out data for those models.
You're picking a model.
We're picking a model, exactly. And what it says is, it is not helpful, the standard statistics, right? It is not helpful to you to include ER as a factor in your analysis.
So that's what you learned.
That's what you learned, exactly. So you learned that it does depend on the microtubules, but it doesn't depend on everything else, okay? On the ER. Sure.
So once we learn the model this way, how do we test the accuracy of the model?
Well, we get more data. I mean, we're out of data at this point, right?
Can you not reverse the identity of the two cells and try to?
Yeah, we actually did that already. right? But that doesn't help. It only helps a little bit.
But it gives you the same result. You find that using informing yourself on the test cell and then.
Yeah.
Right.
Now, I didn't actually tell you, but these are actually calculated by doing that, like swapping, you know, all the possible combinations and so on. So, yeah. But it's the basic idea. Okay, so now here's the thing. Okay, so far that makes sort of sense. Now the task we set for ourselves is to ask these models, these point process models for these 11 different proteins, which we constructed using just microtubules, cell boundary, and nuclear boundary. Can they allow us to tell apart these 11 different proteins? Okay. Now,
Are the 11 different proteins, are all of them associated either with microtubules or ER?
For most of these, nothing was known about whether they're associated with the microtubules. What they are, there are 11 proteins chosen to be markers of 11 different subcellular structures.
So each of them, as far as you know, are in a different cell?
Each of them are different, theoretically. Yes?
And different organelles. Cells,
Yeah.
You're going to get many different distributions of microtubules depending on which phase of the cell cycle. So you've got a dynamic.
Yep. Absolutely. So, first off, I think you can agree this is a difficult task to do by eye. Okay. But if we use those models and we ask for, now we do it differently. We train with one image for all of them, and now we ask, I'm sorry. Yeah, no, I'm saying it right. Train with one image for all of them.
All of what?
All of the 11 proteins. So we train an 11 class classifier using one of the images, and then we ask
So one image from each. Yeah.
one from each, right?
From each, okay.
And then try to classify the remaining one, okay? And we get,
So that means actually 11 images.
so we're going to, well, yeah, so it's a standard multi-class classifier, right? So here are the organelles, and here is the accuracy of getting them, of classifying them. Overall, 83% of those individual images were correctly classified. Now, no human could come anywhere close to being able to do that. Right? These are, and by the way, we learned things like RNP bodies that are not actually organelles. Right? They have been classified by the Human Protein Atlas as vesicles, but they're not. Right? So the point is the models are capturing just from the relationship to the cell boundary and the microtubules this difference between different organelles.
So what you mean is if you measure
These are three.
protein associated with RNP bodies, if you classify it some way, or you measure specific microtubules in the test, then you can go back and accurately find the RNP bodies in the
Well, no, what I was saying is that people classifying those images had said that the proteins that aren't actually in RNP bodies, they had called them vesicles.
Oh, I see.
Because they couldn't tell the difference between RNP bodies and anything else. So that all 11 of these were all classified as vesicles. But the point is we can tell those vesicles by people. They are different spatially. I mean, they're clearly different because the proteins are different, but they're also different spatially.
In ways that you can capture.
In ways that you can capture as in a generative model. And so it's a generative model, so we can actually synthesize a cell with all 11 of these objects in it, assuming they're independent, of course.
Where
All
are
right.
the RNPs in that?
Oh, I forget. I remember who's who. I remember who's who.
Yeah, so I guess sort of philosophical. Well, you're creating a generative model, and it seems to be doing well in discriminative tasks, but the actual model that you generate, or these actual models you've generated, how do we actually know that these are biologically accurate, even if they have, low degree or whatnot?
I mean, hey, welcome to my world, right? I mean, that's
Yeah.
yeah. I mean, that's the issue is, we have data to try to build the model from, right? But then we're going to use the model for something else. Well, the criterion for whether the model is good is going to have to come from that something else.
In other words,
Okay.
you need more data to see.
Well, or for example, we can say, if I build a simulation using model X, does that simulation capture some properties that I've measured by, you know, spectrometry or something else, right? You know, and yes or no, right? Yeah, but that's, of course, a very important question. Yeah?
So, could you elaborate on the point you
Just made, yeah.
about, not the answer to your question, but just before that, in this classification, you assume that all of those proteins are independent. Which,
If I were to generate an image like that, I'm assuming they are independent of each other. Now, I have no information on that because the images I have are just of one of them at a time, right? So I can't measure the correlations between RMPs and, you know, COP2 vesicles because they're not labeled in the same cell.
So that's not there yet.
So that's not there yet. We can talk more about that. That's right. This is a process we're sort of building up from the bottom, right? And we try to add as much information as we can as we go to each level. So we've just added additional information, which is the dependence on microtubules and not dependence on ER. You know, the next experiment when we add something else, right? Okay, a lot of great questions. The next topic is proteins that are not punctate, that are not vesicular, but I'm not going to talk about it because Xiangtao is going to talk about that, which is analysis of changes in protein patterns during cell signaling by T-cell antigen presentation. Won't say anything more about that. What I will talk about, so two more quick things. We were talking earlier about changes during process of differentiation, development, whatever. Very often what we have is a problem in studying systems of development or differentiation, which is that we could try to follow individual cells through the whole process, but if we're doing light microscopy, we're almost certainly going to burn them out. If we try to do it fast enough that we're going to get all the quick changes, they'll be burned out in an hour, and maybe the stuff that's happening over the next 12, 24, 60, whatever hours we get lost. If on the other hand, we only sample every half hour, we miss all the things that happen quickly, right? So this is a classic problem. And the idea is that what we want to try to do, oh, by the way, the alternative is that we just pick representative time points and we take some images and we just make statements about that. Yes?
Wait, I just want to follow up on one thing. Because I don't think I got to actually ask the question before. How many different stages of the cell cycle do you think your generating cell is in at once?
So most of these are almost certainly G1 cells that are imaged.
So your training data actually is held constant.
By virtue of the fact that, you know, in the way that the experiments are done, mitotic cells are probably going to be, you know, knocked off. You know, the cell types have long G1s. You know, so right.
So I can go on.
Yeah. Okay. So, by the way, with enough images, we could have built different models for different cell phases, right? And we've done that too. In that case, we just couldn't. All right. So what we're going to try to do, so we could just do this kind of continuous measure and just hope that we don't burn them out, or we could do representative time points. What we want to try to do is use generative models to bridge those two together. So the idea would be to try to make a movie at high temporal resolution over a long period of time, but make that movie be generated from a model that is learned from example images all the way along. Okay? In this case, we're working with a model system, PC12 cells. You take a cell. This is a very old system. It starts out kind of roundish, and then it differentiates into something that starts to approach neuronal morphology. And we want to know what's the sequence of events that happen there. So we imaged, in this case, distinct time points, at different times, stain with MitoTracker and image. And then we take the cell and nuclear shape and build a diffeomorphic model that you haven't heard about yet, but you're going to. And then we learn one of these point process type models for the mitochondria. Okay? And that's what each time point is going to be represented by a model of shape and the distribution of mitochondria. Now, if we just talk about shape, these are examples of a shape space, again, you're going to learn more about that in a bit, where different cells that have the same shape are near each other and cells that have different shapes are not. Okay? So what it's showing is the progression of cell shapes from small round things to more spiky things as time proceeds. I think I'm going to skip over most of that, and I'm going to just get to this. What we have is, think of each of having lots of cells that we've measured as being represented by some set of parameters in some space. They're each individual cells measured at an individual time. We want to link all of those together. Okay? In linking them together, we have no way of knowing which cell is related to which cell. But what we're going to do is, because we have a lot of cells of both time points, we're going to say the cell from time point 1 that is most similar to the cell at time point 0 was probably kind of like what cell at time point 0 would have become at time point 1. Okay? So we're going to link them up, pair them up. And the way we do that is standard approach, minimum global bipartite matching, right? What pairs of points will give you the minimum overall distance? And so that's what that's illustrating. If we do that, then we can actually learn a kind of dynamics, right? By saying, well, this one that's kind of like that one, well, it changed from this shape to this shape. Okay? So I can learn a model that says that's going to change from that to that. All right? And that's what these are probability density functions of. If you're starting out at this point right here, you're going to move this way. If you're starting out at this point right here, you're going to move this way, et cetera. Okay? Yes?
So to be clear, the experimental setup was having many cells that are differentiating and imaging them all at once at different time points?
At different time points, exactly. Okay. Yep.
And then sort of assuming that any could correlate to any other one in terms of dynamic, right?
Right. Yep. And so here's a synthetic movie of a PC12 cell differentiating, okay, that was created from individual time points.
But are these interpolated between each set?
Yeah, we're interpolating. By the way, I didn't say that. Chantal will talk about that. These models are, you can interpolate in them because they're shape-space models. Right?
So I have a question about this. What are the colors?
Density. Density.
Just density.
Yeah.
So outside of yellow, the density is so low, you just make it.
Yeah, it's just white. Yeah, but yeah, I mean, yeah, yes.
Okay.
All right. So the last thing I'm going to do is very, very quickly talk about using the geometries we get. And I'm going to skip over this. The slides will be on the website. The basic idea is that we start out with some biochemical model like that we could generate with NetGen, write that in SBML, read that in a cell organizer, and it picks out what are the compartments that your model is trying to use. It says, do I have a model of those compartments? And if it does, it generates a new geometry using what's called SBML spatial, merges these two together and generate a script using Cell Blender, and then we can perform a simulation in MCell, for example. Okay? Very, very quickly, if we do that for a very simple biochemical model, we're going to sample different cell geometries from one of these cell shape spaces and do the simulation for each of those sampled cells. And this is the variation in one of the readouts of the model, the dimerized nuclear transcription factor. And what you can see, is there's tremendous variation in that parameter depending on which geometry it is, right? So these are for different geometries. Okay? That's all I'm going to say about that. The point is that we can take these, use them as inputs for generating geometries and for generating simulations, and look at how the behavior of the model is affected by the geometry of the cell. Okay? In a much easier manner than if we went in and had to hand draw thousands of cells or hand segment it, hundreds of thousands. So, okay? So that's sort of the end of my story. That the idea is that these generative models provide better ways of comparing across experiments, labs, instruments, and so on. They provide good geometries for cell simulation. By the way, they can also be used for testing image analysis algorithms. I didn't talk about that. And the idea is to put all of these kinds of things together through this framework of a generative model. Okay? So that's it. And if there are any more questions, I'll take them. All right.